Okay, I hope that you can hear me. This is your notes on maintaining homeostasis, which is actually a little bit of a hodgepodge of a couple of different chapters because really homeostasis is something that is happening all the time in all living organisms, in every single tissue, in every single cell. And so they sprinkle a little bit of homeostasis in lots of different um, chapters in your textbook. I'm trying to condense it into one because the main thing I want you to understand is I want you to understand how we use positive and negative feedback loops to help maintain homeostasis. That's my goal really of this particular um, set of notes. So let's move that down. So just to remind you kind of the organization of your body or of any tissue, living organism, is you start with the basic unit, the cell. Okay, and then remember the cell is highly organized and every part has a different function. And then the cells combine to form tissues. And then the tissues are going to work together to operate some type of organ. So for example, you have cardiac cells, and those cells form your cardiac muscle tissue. And then all the tissue within that cardiac muscle is going to form together the entire heart, the organ. Okay, and then organs are going to work together to form an organ system. So your heart is an organ, but it is going to work with your entire circulatory system to perform a specific function, which is to pump blood. The organism's homeostasis is its stable internal environment. So we usually have a set point for everything, and we want to stay right around that set point. So think about body temperature, blood pressure in us. Those are all things that we want to keep at a normal range. And all living organisms are that way. They have a set point for most things, and they want to stay kind of right around that set point. And it's largely dependent on the work of the individual cell. It all comes down to how the cell is actually functioning and what it is doing. And if there's a problem with the cell function, there will be a problem with the tissue, which will be a problem with the organ, which will be a problem with the organ system. It's a domino effect, and it all goes back down to each individual cell and how it's functioning. Homeostasis is a steady state or a maintenance of an internal balance. Animals are able to maintain a relatively constant internal environment even when the external environment changes significantly. That's actually pretty cool. What kinds of homeostasis do humans exhibit? Sorry, I'm recording a video for AP. Hang on, let me see if I can pause it.
This thing, because I'm too lazy to fill it out. I'll put it right there.
Oh, okay, sorry about that. So let's talk about a little bit about animals and how some of animals are what we call regulators and some are conformers. So regulators use internal mechanisms to control internal change in the face of an external fluctuation. For example, if the outside temperature goes up to 100 degrees, your body's set at 98.6. You're still going to stay about 98.6. And the flip side, if it goes down to 0 degrees Fahrenheit, your body is not going to go to 0 degrees Fahrenheit because you have the internal mechanisms to regulate that body temperature. So those are what regulators are. They're anything that's going to regulate. They're not going to just conform to the environment. They're going to regulate their own internal. A conformer is actually going to allow its internal condition to change in accordance with the external changes in the variable. Many marine invertebrates let their internal solute concentration conform to relative stable solute concentration or the salinity of the ocean environment. So there's an example of a conformer. They are not going to regulate their own internal and be different than their environment. They are going to conform to that because that makes the work that they have to do a lot less. So you're going to find a lot of saltwater organisms, their internal salinity solution is about the same as the ocean. And if you take them out and go put them in a freshwater pond, it's not going to work very well. And this is just showing the same thing here with temperature. A lot of fish and reptiles are like this. As the temperature goes up, they actually change their temperature and they conform to that outside environment. A river otter, which is a mammal, is going to do something completely different. It's going to regulate its temperature. So even though the outside temperature is going up, they're actually still staying within this margin here. So here's a non-living example of a feedback system, a home heating system. So here you have your set point. You set the room temperature to about 20 degrees Celsius, which is, 70-ish, maybe a little less. Okay, and the stimulus is the room temperature increases, and the response is your heating unit stops. Okay, so then your room temperature is going to go down because there's no longer being heat emitting out of it, and it's going to get back to that set point, and then it's going to get lower or around that set point, and then it's going to temperature is going to increase. The heating is going to stop, and it's just going to keep kind of going in that same pattern here. So room temperature decreases, heating starts, room temperature increases, going back to the set point. And then it'll stop, and then so it's just a constant flow of stopping and going, turning on, turning off. You'll notice this in winter. You'll notice your furnace kicks on and then it kicks off, and the reason is because it's all trying to stay around this set point. This is an example of negative feedback. It relies on a control mechanism that reduces the stimulus. Example: if you exercise vigorously, you produce heat. This increases your body temperature. The nervous system detects that, triggers sweating, and as you sweat, the evaporation of moisture from your skin cools your body, helping return your body to its set point. So the idea with negative feedback is that whatever the control mechanism is going to do, it's going to reduce the stimulus, going to try to get it back to that set point. Now, that's different than positive feedback loops. Positive feedback loops amplify rather than reduce the stimulus. So this doesn't play a huge role in maintaining homeostasis, but it typically drives a process to completion. Okay. Example: during childbirth, pressure on the baby's head against the receptors near the opening of the mother's uterus stimulates the uterus to contract. Contractions result in greater pressure against the opening of the uterus, heightening contractions and causing even greater pressure until the baby is born. Notice in this situation, you're looking at some keywords: stimulates, greater pressure, heightening contractions, greater pressure. Everything is moving towards an amplified stimulus or an amplified response to a stimulus. Whereas negative feedback is always going back to the set point. It's going to do whatever it does to regulate or go back. Can you think of other examples of negative and positive feedback? If you do, write them down as you are thinking of them. We're going to hear 8 examples in class because you are all going to create a presentation over an example. Homeostasis moderates but doesn't eliminate changes in the internal environment. Additional fluctuations might occur if there is a normal range rather than exact set point. Sometimes the set point is specific, sometimes it's more of a range. Set points in normal ranges can change under various circumstances. Regulated changes are essential to normal body functions, such as huge change in hormone levels during puberty. So just because there's a set point doesn't mean that set point's the same always. Sometimes that set point changes, and it can change drastically. Puberty is a good example of where that set point changes pretty drastically. Your hormone levels will change significantly. And still, there's going to be some major changes, but we're going to now readjust that body to a new set point. Circadian rhythms is a set of psychological changes that occur roughly every 24 hours. For example, your body temperature actually fluctuates a little bit depending on the time of day. Melatonin concentrations, that's going to be what helps you kind of get sleepy. Light and dark in the environment, those are things that you're triggered by the light and the dark. This is where you get things like jet lag, because even though you've changed times drastically, your body is still on a 24-hour cycle, and it's used to performing upon that 24 cycle. And when you go to a new country where that 24 cycle is flipped or upset a little, you're going to experience a little jet lag. You're going to experience a hard time getting back to it. Now, if you were to stay there a month, you would set your circadian rhythm to that particular environment, and then when you came back, you would have trouble adjusting again. This shows a graph of body temperature as you time of day. It actually goes up about the afternoon, goes down, and your body temperature is the lowest in the evening, or sorry, early morning, middle of the night when you're sleeping. And then melatonin is actually highest when you're sleeping. Um, some people take artificial melatonin as a sleep aid because it's naturally secreted in your body, supposedly it's a little safer um, than taking a prescription Ambien or something as a sleep aid. And this tells you like your heart rate is lowest when you're asleep, and then you have the fastest reaction time kind of in the evening. So you'll notice that your body has a circadian clock. It has a clock it is set to. And if you upset that by going somewhere where the clock is different, you're going to experience some problems. I really struggle with this word. Acclimatization is acclimatization is the gradual process in which an animal adjusts to changes in its external environment. Example is moving to higher elevations because there's less oxygen. It's going to take you a while to adjust to that. You'll have increased blood production of red blood cells to carry oxygen. It'll raise your blood pH, causing kidneys to alter their functions in order to lower the blood pH once again. So you're going to experience some changes in your body to readjust to that new set point. Here's a couple of examples that are really important in homeostasis that I'm going to highlight. One is thermoregulation. This is the process by which animals maintain an internal temperature within a tolerable range. There are two types of organisms, endothermic and exothermic. Not ectothermic. Endothermic are warmed mostly by heat generated by metabolism, birds and mammals. Okay, so that's how we are really have a good set point. We use our own body internally to regulate that. Ectothermics gains most of their heat from the external sources, which is why you also see that their temperature changes more. They're more of a conformer than a regulator because their heat comes from an outside source, which may or may not be there every day. A walrus is an endotherm, and a lizard is an ectotherm. You will find that lizards and other ectotherms tend to like to be in the warmth. They seek out the sun because that's where they gain their heat from. Thermoregulation depends on an animal's ability to control the exchange of heat with its environment. They exchange heat by means of radiation, evaporation, convection, and conduction. Let's show a little bit of that real quick. So radiation is the sun. Evaporation is breathing, like sweating or breathing harder. Conduction, they can get on something that's warm. And convection is air movement over their body. Insulation reduces the flow of heat between the animal and its environment. Think about like polar bears, they have a really thick fur coat, or even your dog probably gets a thicker coat in the winter, especially if it's an outside dog, because that hair creates a really good barrier to keep heat in. Feathers, fat does the same thing, blubber. There's a really good, if you don't ever watch, if you got Netflix, if you ever watch The Magic School Bus, I know it seems so silly, but there's a great episode on this where they actually get like flubberfied, and the kids get all this blubber on them, and they realize just how warm that is. Because they realize that, like, you know, seals and walruses and things in the ocean have lots of blubber, and that helps keep them warm. Blood flow regulation, altering the amount of blood flowing between the body core and skin, is a means of regulation. Vasodilation, this is when you have a widening of your blood vessels, increasing blood flow and warming the skin. Vasoconstriction, this reduces blood flow and heat transfer by decreasing the diameter of superficial blood cells and keeps them from overheating. So you'll notice that you might have some vasodilation or vasoconstriction in your hands because you have lots of little blood vessels there in response to the environment. If it gets really cold, you might notice that they get really small, or you're not as pinkish in your hands, and that's because they're constricting and they're trying to reduce the blood flow and keep that that blood at your core to keep you warm. When you're really warm, you might notice some pinkening, and that's because they're widening up and allowing that blood flow. And you might notice that, sorry, I was concentrating on something else for a minute. You might notice that if you're cold, you're going to warm the skin, and they're going to dilate and kind of open up. So countercurrent exchange is the transfer of heat or solids between fluids that are flowing in opposite directions. Examples: artery and veins are located near each other so that they can exchange heat. Here's an artery, here's a vein, and they will actually exchange heat. Warm blood and then cool blood will get heat transfer from one to the other, and so they kind of lay next to each other so that they can exchange heat among them. Behavioral responses might be migration or hibernation. Think about bears, they hibernate because it's too cold. Migrating, birds migrate when it gets too cold. Adjusting metabolic heat production is shivering or moving increases your muscle activity. You probably noticed that if you get cold, you shiver, and that's actually your body's response to the cold, trying to warm you up. And then moving around increases your muscle activity, which will then keep you warmer. So you probably know that. If you stand at a bus stop on a cold morning, if you move around a little bit, you seem a little bit warmer. So this kind of goes to your, we talked about the house and the heating system in the house. This is like your heating system for your body. So your temperature is supposed to be right around normal. And then if your body temperature increases above that, or for some reason the external environment tends to warm you a little, your brain is going to sense that, and it's going to cause your blood skin to dilate, and you're going to sweat, and then your body temperature is going to decrease. And then if that does that, your body temperature decreases back to a normal set point. If for some reason it decreases too much, your brain's going to sense that. Your blood vessels are going to constrict, trying to conserve that heat. And then you're going to shiver. And then your body temperature is going to increase from that, and it's going to go back to the normal set point. And then it's just a continuous cycle among those things. Osmoregulation is another important mechanism of homeostasis. It's based largely on the controlled movement of solids between internal fluids and the external environment. So think about that fish again. Recall from osmosis, if water uptake is excessive in animals, cells will swell and burst. Okay? Think about your cell. If it's in an environment where there's more water, say there's 90% H2O, so only 10% solute, and the cell has 30% solute or 70% H2O. Water is going to move in, it's going to swell up, and it could burst. If water loss is substantial, they will shrivel and die. So just the opposite situation here. If the outside has a lower concentration of water, so for example, this is 90% H2O, and the outside is only 70% H2O, then water is actually going to flow out of the water and out of the cells, and it's going to shrivel up and die. This is the reason why. This is the reason why we don't drink pure water, 100% H2O, or you don't want to just drink distilled water, is because it could actually, or too much water even, and people that run races and stuff sweat a lot and they drink a lot of water. If you just drink pure water, a lot of times that will cause your brain cells to swell and burst, because your cells are not 100% water. And this is an example here where your cells can actually shrivel up and die. I'm trying to think of a really good example here. Too much salt in your diet, you could actually damage your cells by not giving them the proper amount. Movement across a selectively permeable membrane occurs when two solutions are separated by a membrane, differ in their osmotic pressure or their osmolarity. Osmolarity just means their osmotic pressure or the difference in the amount of water concentration. So if you've got two solutions that are separated by a membrane, and this is 90% water and this is 70% water, you're going to see water go from a high to low environment. Osmotic pressure is basically the difference in 90% and 70%. So here is what we're talking about. So this is the hyperosmotic side because there's a higher solute. Hyper means higher. So if you are hyperactive, you are more active than other kids. So hyper is referring to the solute. the thing that is not water. Okay, so that means there's lower free water available. In a hypoosmotic side, you got a lower solid concentration. So hypo means lower solid. Okay, hyper means more solid. And then that means there's more free water available. And so that water is going to go to the side that is hyperosmotic. Water always flows hypo to hyper. Remember that, because you are going to have osmosis questions on your AP exam. Water always flows hypo to hyper. If the two solutions separated by a membrane have the same osmolarity, they are isoosmotic, or you've probably heard isotonic is another way to put that. And no net movement will occur. That doesn't mean no movement will occur, it just means no net movement. There will be particles going from right to left, equally going left to right. If they are not the same, the solution with the greater solute concentration is hyperosmotic, and the more dilute solution is hypoosmotic. So here's what you want to know if you've got an osmosis question on your AP exam. You want to identify which solution is hyperosmotic and which one is hypo. Once you can figure that out, you know water moves hypo to hyper. Now, the solute will only move if the membrane is permeable to it. Okay, so you have to identify that. You have to figure out what the solute is the membrane permeable, and then the solute is always going to move high concentration to low concentration. Water flows hypo to hyper. An osmoconformer is to be isoosmotic with its surroundings. An osmoregulator is going to control its internal osmolarity independent of that of its external environment. All osmoconformers are marine animals. Think about that. They live in an oceany water. It's going to be more advantageous for their cells to conserve energy if they can just be like their environment. So that means they're not going to gain or lose water too drastically. They're going to pretty much be the same because they are going to be that isoosmotic. The exchange is going to be pretty even. Most animals cannot tolerate substantial changes to their external osmolarity. So osmoregulation in marine fish, if you look at this, it kind of talks you through how that's going to happen. They gain water and salt ions from their food and from drinking seawater. They're going to excrete some of that from their gills. They're going to have some water loss from their gills and other parts of their body surface, and they're going to excrete salt ions in small amounts of water in the urine from the kidney. They're taking it in, they're excreting it, but it's all going to be about the same. An osmoregulator is like a freshwater fish. It's going to be a little different. They're going to gain water from food and in the water. They're going to uptake salt ions by the gills, and then they're going to have osmotic water gain through the gills and other parts of the body surface, and then they're going to excrete a little bit from their urine. Okay, so they're not going to be really like their environment. They're going to be more in terms of they have to regulate it. So they have to take in or lose in order to keep an internal homeostasis. In vertebrates, kidneys function in osmoregulation. That's one of their jobs, is to control that. You'll probably notice that if you're not drinking a lot of water, that your urine is very, very yellow, and that means that it's very, very concentrated. There's not a lot of water in it because if you urinate all that water out, you're not going to be maintaining that homeostasis anymore. You'll notice if you're drinking a lot of water, your urine is actually almost clear. It can get to be a little bit yellow, but not very much yellow. That's actually a really good measure of whether you are dehydrated or not, is looking at your urine. If it's really yellow, you probably need more water because your body is conserving water and just putting out the urea. And if you are a very light yellow to almost a clear, then you're drinking plenty of water. That's where we actually all should be. And if you want, you can refer to chapter 44.4 on kidney function. So this is really how the kidneys work here. Your kidneys, you've got two of them. They're like bean shaped. They're actually kind of to your back. So if you have a kidney infection, you'll actually feel back pain instead of abdominal pain. This is the structure of the kidney. If you get up close to what is called a nephron, this is where all of the exchange is happening. It's taking in and it's releasing. This is something that was a lot of fun to learn when I was in physiology. And I'm saying that very happily. You've got lots of arteries and veins here. Remember, they're going to exchange some temperature. this is your loop of Henle, and this is going to come down like this, and this is your descending and ascending loops, and this is where a lot of that exchange is going to happen. Okay, you're going to exchange liquids. Notice here they talk about NaCl, nutrients, water, and these are just going to flow in and out of that membrane in these kidney tubules depending on what you need. Do you need to lose water? Do you need to gain water? There's going to be some active transport dealing with the salt, and then there's going to be some passive transport dealing with the water, because the salt has to go through the sodium potassium pump. It can't just diffuse. It's too large. It's a larger compound. And then so here it's talking about a measure of your osmolarity as you go along the way. You don't have to memorize this, but knowing that as you're going through this loop of Henle, part of the kidney, there's going to be a change in the osmolarity, and that's dependent on what do you need. Do you need more water? Do you need less water? Do we need to get rid of the water? Or do we need to conserve it? So that was a quick introduction. You don't have to know anything really about the kidney function. I just want to introduce that is one of the jobs of the kidney is to regulate that water for you.